When I'm done, you're going to want to pay very close attention because you're going to get very exacting details on how to make a couple of cocktails tonight. So if I haven't met you, I think I've met everybody here. My name's Mitch Drum. I'm the director of the Research Institute for Children's Health. And we are here tonight to thank you for being supporters of what we're doing. And we also like to have fun, so that's another underscore of the evening. But I think before I do anything else, I need to thank a number of people who were absolutely instrumental in making this happen. And I'm going to start with the ones why this happened, this type of event. So Tom and Mary Beth Breckenridge, right here, waving the hands. So these are longtime friends of ours down in Brecksville. And they've been serving on our advisory council and decided that there are friends of theirs that they used to work with, John Mangles and Andrea Samakis. Where are they hiding from me? Right back here. So during COVID, John and Andrea decided they needed a hobby, and so they started working on making wonderful cocktails. And so Tom and Mary Beth asked them if they would come to their house and have a little fundraising event for us in their basement. And they did, and it was such a wonderful, everybody had such a great time. And then I remembered there's another friend of mine, this tall guy here, Francis Collins, who seemed to have done the same thing, that during the COVID times that he tuned up his cocktail making abilities. So we thought, okay, let's do that and see if he'd be willing to come to Cleveland and join us for the event. And he did. So anyway, so I want to thank John, Andrea, Mary Beth, Tom, Francis, and Francis's wife, Diane, for making this happen. But then also folks at Case Western who have been instrumental in this. Rebecca Mason, Heather McNally, Emily Corona-Lusher, and is Kathy Malone, where's Kathy? Okay. So Kathy has helped us with every event that I've ever been involved in. And then our AV group, so Tim, Dwayne, Noah, did I miss any? Oh, Davey, where's Davey? Okay, that's why I didn't see him. Yeah. So anyway, so these are all the folks that are making tonight possible. So thank you for that. Now, you're probably wondering, okay, in addition to having fun and having drinks, why are we here tonight? So we started about 10 years ago, not about 10 years ago, we started an effort to apply what we've learned on genetic diseases, in particular one called cystic fibrosis. We said, well, we've figured out a lot of the steps to help make new therapies for that disease. And the steps are not all that different for many other genetic diseases. So there's another 6, 7, 8,000 genetic diseases out there. We said, we've got all the tools here to do it. We just need the funds to get those things off the ground. And that's what the Research Institute was designed to do. I haven't asked him to talk about it, but if he just so happened to, Dr. Collins was the person that led the genome effort. So you probably all have read that the human genome was sequenced, and we now have this incredible map of all the genes across all of our chromosomes. And this was an endeavor that took, and he can correct me on my facts here, but roughly 13 years and about $3 billion to do. And now we can do that depending on where you do it, but more like $500 or so depending on what level of clinical involvement you have in things. But in about 24, 48 hours, you can do the same thing that took $3 billion in 13 years to do, right? So that means you have a technology now that can move us so fast. And it's now the bottleneck for us in the labs is not finding the genes that cause the disease, but it's how do we fix them. And that's what we're working on, trying to apply some of these new technologies. You may have read of something called CRISPR. That's the kind of thing that we're doing now. We can actually go into the cells, fix the mutation that causes them. Just to give you, again, magnitude of the issue, your body's probably got somewhere in the neighborhood of 20 million cells or so. So, and they all, if you have a genetic disease, every single one of those cells has it. So, quite an engineering feat to try to figure out how we're going to fix those. So, anyway, I won't go on any longer, but just want to also let you know, if you have any desire to see what we do, we invite you to come to the lab. We'll take you through and show you the different facilities we do. I think it's been one of our most effective, impactful things, right, Deb? To show you, so you can actually see where the money goes and make it less abstract and get a sense of what it's all about. So, so right now, if you don't mind, we're going to take our seats here. And we will start. John Mangles is going to lead you through the construction of the concoction that will be the first drink. And he's an awesome teacher. So he was a science writer for the Plain Dealer. He understands the steps. And then after the first one, while we're clearing off and getting ready for the second one, we're going to do, I get to do a little interview of Dr. Collins. So this is, I can't call it payback, but it's a different kind of relationship. So he took pity on me 40 years ago. We both got to the University of Michigan about the same time. He is a new professor, I as a new graduate student. We met at the department picnic, and he let me do a rotation in his lab. And so I have what I think is the distinct honor. I was the first graduate student in his lab that he had, and we've been wonderful friends, at least my perspective. We've been wonderful friends ever since. We've gotten to watch each other's kids grow up, and just been a fabulous journey together. And if you don't know, if you haven't watched the news, if you've had your head in the sand, you will have seen him many times. So whether it's on the news, if you're Colbert fans, he's been on, I think, 6 times on the Colbert Show. So, and is a very, very strong advocate, not just for science, but just for truth and honesty as well. And I think that's going to be our focus more than anything else right now. We can talk the science if you want to, but I think it's how do we bring civility back to our world. So anyway, maybe drinks that's a good segue to drinks. So, John? Okay.
Oh, thanks. I'm good.
Oh, okay.
Okay, everybody. Who's ready to make some cocktails? Yeah? Well, welcome. Thank you all for being here. Hope you can hear me. I am Dr. Cocktail. And I want to thank you all for being here tonight. It's great to see all of you and to be able to support a great cause, Mitch and the Research Institute for Children's Health. Just an amazing place. And I hope all of you are able at some point to go on a tour. It's an impressive place, and you can really see the things that Mitch and his team are accomplishing. And it's so great to have Dr. Collins here. I mentioned to him many, many, many years ago when I was still a science writer before I became a degenerate cocktail maker. I actually was at the news conference where the Human Genome Project's results were announced and met Dr. Collins back then. He doesn't remember it, but it was a big deal to me. So it's good to see him again, and good to see all of you. Give yourselves a round of applause. I mean, you guys are a great group, and you're supporting a great cause. Thank you, thank you, thank you. So we are going to dive into the world of tiki tonight. We're going to learn some tiki history. We are going to learn a little bit about cocktail chemistry and cocktail theory, and a little bit about the mechanics of mixology. This is going to be a fun class. There won't be any tests. I promise my lecture won't be dry. Get it? There you go. Thank you. Yeah. Smart audience. This is going to be a very hands-on class. I love to teach classes like this. All of our ingredients are on the table, and I'm going to talk you through all of the steps involved. And the great thing about this is at the end of the night, you get to keep your recipe card, your glasses, and your mixing equipment, so you can go home and really impress your friends and recreate what we do here tonight. So, everybody ready? All right. Let's talk a little bit about tiki history. We're going to go back in time, 100 years almost. 1933. What's happening in 1933? Anybody know? Prohibition ends, right? 13 years of no alcohol in the United States is finally over, and people are thirsty. They want to drink. They're tired of bathtub gin. They want some good stuff, right? So, in Hollywood, California, there's a young entrepreneur, a 20-something-year-old guy named Ernest Gantt. And Ernest capitalizes on the availability of alcohol once again, and he says, I'm going to start a bar. He'd never done anything like that, but he was inspired. Ernest had spent the last few years sailing in the South Pacific and the Caribbean with his grandfather instead of going to college, which is what his parents wanted him to do. And he and his grandfather had collected a bunch of stuff, like a bunch of bric-a-brac, a bunch of tiki-themed stuff from the islands. So Ernest comes back to Hollywood, and he finds an old tailor shop, and he converts it into a bar. He decorates it with all the stuff that he collected on his travels with his grandfather, and he calls it the Beachcomber Cafe. And he reinvents himself. He's no longer Ernest Gantt. He is Don the Beachcomber. There's Don the Beachcomber. Don the Beachcomber wears straw hats and leis and loud shirts, and he begins to make these fabulous drinks. He actually invents a whole different kind of cocktail. He uses rum, which comes from the Caribbean, so he knows it well. And he makes these crazy concoctions with names like Cobra or Missionary's Delight. And people love it, right? So it's the middle of the Depression, and people are looking for some kind of escape from the troubles in their lives. So they hear about this little hole-in-the-wall bar with its tiki theme. And also remember, this is the Depression, so no airlines, no fancy travel. To go on a cruise at that time would have cost you a lot of money. So most people have never gone anywhere except for, around where they lived. So the idea of going to somewhere that is exotic really appeals to them, right? This place takes off like wildfire, draws huge crowds, wildly popular. Well, up in Oakland, California, there's another young man who's also an entrepreneur. He's got a bar at this point. It's a hunting lodge themed bar called Hinky Dinks. And he hears about Don the Beachcomber and Beachcomber Cafe. And he decides, well, I got to go check this out. So he drives down to Hollywood, and he starts to hang around, and he starts to take notes about everything he sees, to the point where Don the Beachcomber gets a little ticked off about that, because he can tell what Victor Bergeron is up to. And he finally bans him from the bar. But Victor has got enough notes, so he goes back to Oakland, and he starts trading for stuff that he can decorate his bar with. His wife knows that he likes to trade stuff. She actually calls him Trader Vic. That's her nickname for him. So Trader Vic changes the name of his bar to Trader Vic's. It's a tiki bar, and tiki takes root in Oakland as well as in Hollywood, and it becomes wildly popular there. And from there, it spreads across the country. Peak tiki is in the 40s, 50s, 60s, and somewhat in the 70s. So it's going around the country. Ohio is no exception. Cleveland is no exception. The Club Zombie opens in the 40s. The Kon-Tiki opens up in the late 50s, early 60s. It's in the Sheraton Hotel downtown, a hotel that's still there today. In Columbus is one of the greatest tiki bars of all time called the Kahiki. You can see it's shaped like a war canoe from Polynesia. It has jungle, tropical rainforest themed areas and lightning and thunder and a giant fireplace shaped like a tiki god. My lovely wife, Nurse Andrea, who grew up in Columbus, used to go to the Kahiki in her youth. But like all fads, tiki fades after a while. So by the time the 1980s and the 1990s rolls around, tiki begins to disappear. And if you were to go to a bar in the 80s or the 90s, you probably wouldn't find a tiki drink on the menu. But the craft cocktail movement occurs in the early 2000s and 2010s. It's back to the basics in cocktails. And part of that movement embraced and revived tiki. So tiki comes back, but it's not quite the old tiki. I mean, of course, the classics are there, but these new craft cocktail mixologists decided they want to tinker with things. So they're swapping out ingredients, they're inventing whole new drinks. New tiki bars open. There's a great one in Cleveland called Porco's on West 25th. If you haven't been, I encourage you to go. It actually has some of the memorabilia that came from the old Con Tiki from the 60s. And they have wonderful classic and new tiki drinks. So tiki's back, right? We are going to talk about and celebrate these two eras in tiki history, the classic era and the revival. We're going to start with the classic era in this first session. And in the second session, we'll go to kind of a more modern take on tiki. The cocktail we're going to make is a variation of something called the Blue Hawaii. The Blue Hawaii was invented by a lovely guy named Henry Yee. That's Henry Yee at age 100, kind of crazy. But in 1957, Henry Yee was the bartender at the Hawaiian Village Hilton in Honolulu. And in walks a sales rep from a Danish distillery. And this sales rep has this crazy blue stuff. It's blue curacao. What is blue curacao? It is a Caribbean liqueur, an aperitif. It's made from a fruit that grows in the Caribbean on the island of Curacao. And this fruit is called the laraha. And if you peel the laraha and you dry the peel and you soak it in alcohol and you put spices around it and let it kind of absorb all that, and then color it with blue food coloring, that's kind of the funny part of it, you have blue curacao. So the sales rep says, Henry, we're here in Hawaii. Can you make a drink with this product that I really would like to sell to lots of people? And Henry is up to the challenge. So Henry makes the Blue Hawaii. The Blue Hawaii has rum, vodka, pineapple juice, sweet and sour mix, which was, by the way, was invented by Don the Beachcomber. And it's very popular. People love it. It's colored with blue curacao, of course. So it's this tropical blue color. We are going to make a variation of that. There are always variations of tiki drinks. I should say Henry Yee named the Blue Hawaii after a song, and it's not the Elvis song that everybody remembers. It is Bing Crosby's version of that song. Bing Crosby actually recorded Blue Hawaii back in the 30s in a movie called Waikiki Wedding. Elvis remade Bing Crosby's song, a lot more popular, but that is a fun fact. So we are going to make the Blue Hawaiian. Blue Hawaiian is a variation of the Blue Hawaii. It's a little bit different. It still has the rum. It still has the blue curacao, of course, to give it this lovely color. It still has the pineapple juice, but instead it's got cream of coconut and lime juice. So that's what we're going to be up to here. Next slide, lovely assistant. Let's talk a little bit about how we're going to make this drink. So when you're making a drink, if it has fruit juice in it, you need to shake it up. Don't ask me why. You just do. Trust me on this. So we're going to use shakers, and you're going to learn how to use shakers. There are two kinds of shakers out there. There is the cobbler shaker, which some of you may have at home. It's a great shaker. It's got a strainer built into it. When you shake a drink, you are mixing the ingredients. That's one thing. You are aerating. You're adding air, right? So that gives it kind of a nice fizz and froth. You are chilling it because this will have ice in it, and it's metal, so it's automatically going to quickly chill it. And it's going to dilute it a little bit because the ice is going to melt a little bit. And you want a little bit of dilution in your drink to kind of cut the sharpness of the alcohol. So cobbler shakers are great, but there's a drawback to the cobbler shaker. When you put ice in them and shake them, this top gets stuck, and it's really hard to get off of. And I can't tell you the number of times I've been like that, trying to get it apart. And if you're in a bar, time is money, right? You can't be behind the bar trying to get your shaker undone. So most professional bartenders use something called a Boston shaker. That's what this is. You guys have them on your tables. You've got, it's the black shaker like that, right? And in it, there's a Hawthorne strainer. I'll talk to you about the strainer in just a second. You've got a metal side and you've got a glass side. I've got two metal sides, same thing, just a slightly different design. The strainer is so that when we mix up our ingredients and shake them up, and I'll talk to you about how to shake them, then you're going to strain your drink into your cocktail glass. And you do that by putting your Hawthorne strainer over and holding it like this, and we'll pour it into our tiki glasses, right? All right, so the first thing we're going to do is we're going to make a garnish, because every tiki drink has a good garnish. What does blue Hawaiian look like? It looks like the ocean, right? So we need an ocean-themed garnish, don't we? Yes? Come on. This is a participatory class. All right. So we are going to make a garnish first. Look on your tables. You'll see a banana, a half a banana, right? Pull your banana out. I wish I had one of these to demonstrate. Oh, I do. Thank you. All right. Terrific. So let's take our banana. You'll see that the stem on the banana is cut, right? Does it look like anything to you guys? Well, just wait. We'll get there. All right. So on the end of your tables are little containers of cloves. You see those? Everybody see that on the end of their table? There should be one on each table. Just give it a smell. You'll know where the cloves are. All right, we're going to take 2 cloves each. Everybody take 2 cloves out. Pass them around. Take your 2 and pass them around. Everybody raise your hands when you're clove ready. Everybody ready? All right. They're getting there. All right, take your clove. Take 1 clove. And on one side of your banana, push it in. And do the same on the other side. 1 here and 1 here, right? Got it? All right. Our creation has eyes. Now, on the back of the banana, there should be a slot, a little cut, right? You see it? Okay. Do we have the fronds, pineapple fronds? Assistants? We've all got them? Okay. Take your pineapple frond and stick it in your banana in the slot. Might be a little hard to get it in there, but you can do it. Dr. Cocktail's having trouble getting it in there, but you guys will do better than Dr. Cocktail. All right. Kind of, sort of. You know what? If you can't get it all the way in the slot, that's okay. We're not perfect here. We're just going to have fun. All right. Then take your Luxardo cherry. You have a container with 2 cherries in it. Take one of those out and put it in the mouth of your creature. Got it? So put it right in the mouth, right? Everybody see that? What does it look like? Who's got it? Come on, somebody. There you go. Yeah. Okay. All right, we've got our garnishes. We're ready to go. We're going to make the cocktail, right? Set your garnish aside for a second. Lovely assistants, ice time. We got ice on the tables? We got ice on the tables. Okay, take your tiki glass and fill it about halfway, about up to here, with ice.
With the crushed ice.
Crushed ice. Yep. You know what? Whatever's on the table is fine. Fill it up about halfway or a little bit higher, right? Let me know when you're filled up with ice. Okay. Everybody got your ice pretty much in your glass? Yes? All right, take your garnish, take your dolphin, and position it on top of the ice. Poking its little head out of the glass. There you go. That's great. Perfect. All right. Now, we're going to make the drink. Take your shaking tin, and in the big one, we're going to add our ingredients. So all the ingredients are on your table. You can put them in.
The top of your shaker is inside your shaker.
Yes, take the glass out of your shaker. There are two halves. You've got the glass half and the metal half, right? So in the metal half, in the big one, put all of your ingredients in. All of them, just pour them right in. If you were behind a bar and you were measuring these and mixing them, you would put the cheap stuff in first and the expensive stuff, meaning the booze, last. Because if you messed up with the measure, you wouldn't waste the whole drink, right? But we don't have to worry about that here. So dump them all in there. Everybody wave your hand when you're done dumping all your ingredients in the big shaker tin. There you go. You guys look like mad scientists. You are great. We're going to put you to work in Mitch's lab, mixing chemicals. All right. How are we doing? Everybody got their ingredients in the shaking tin? All right. So now we're going to put ice into the smaller tin, the glass part, right? So fill it up to, about yay there somewhere. It doesn't really matter which ice you use. I mean, ideally it would be crushed ice, but it doesn't matter. You're going to fill it up about there. Not quite to the top, just a little bit below the top. Okay, who's ready for the exercise? Ready? You guys warmed up? Okay. Here comes the fun part. We're going to put these two halves together, and we need to do it a little bit carefully. So take the big one that's got your ingredients in it, hold it in one hand. Take this half, which has the ice in it, and we're going to tip the ice into it and put it together. And it goes together kind of on an angle like that, right? And we're going to give it a tap to make sure it's together. Kind of on an angle. I'm going to walk around and show you guys. So like that, right? You'll see a little bit of a gap on one side, kind of tilted like that. Everybody see this? Kind of on an angle, right? Give it a tap. Everybody see this? All right, now we're going to, what? Don't shake it yet. Don't shake it yet. You're going to be sorry. All right, so the thing about Boston Shakers is you've got to have a grip on them to do it right. You don't want it to come apart because you will be wearing your cocktail, not drinking it. So we've got it together. We've tapped it. We're going to hold it on the bottom. We're going to hold it on the top. And we're going to do this, right? Give it a shake. Come on, give it a shake. Come on, you can do it. 20 seconds, 20 seconds. Keep going. Let me hear that ice. Give me a workout. Show me those muscles. Come on. There you go. Nobody's wearing their cocktail yet, right? You're all doing it right? Okay? You got it? All right, we're there. All right, now we're going to take it apart. And this can be a little bit of a tricky thing because they tend to kind of get a little stuck together. So the way we're going to do this is we're going to kind of pinch the bottom one, the metal one, and pull. And you can kind of tap it to drain it. Everybody got theirs apart? Can you get it? Anybody need help? You can do it. Give it a little bit of a squeeze here. Let's get that vacuum broken a little bit. Wow, you really got it. There you go. Okay, we're gonna tap it a little bit. There you go. Everybody got theirs apart? Okay, take your yes? Anybody having problems? You got it? Everybody? All right. Take your Hawthorne strainer, spring side down, put it on the top of your strainer, your big glass, right? You're going to hold it with your index finger, and we're going to pour it into our tiki glass. Oh, that's beautiful. That's beautiful. Look at the color. That's great, you guys. All right. We're going to add a little bit more ice. If you want, you don't have to, but you can add a little bit more ice. How's your dolphin doing? Did it sink or is it in there? Okay. Put your colorful straw in your drink. Everybody find their straw on the recipe card? Okay, let's see how we did. Try it out. What do you think? What do you think? Are you guys professional mixologists now? I hear lots of happy chatter, so I'm assuming it tastes pretty good. You can certainly eat your banana and your Luxardo cherry if you'd like. So, how are we feeling? Are we all tiki professionals now? All right. All right. Give yourselves a round of applause. Good job. We are going to take a little bit of a break here and move over to the second stage where Mitch and Francis are going to speak to each other and speak to all of you. So take your drink with you, and we'll reconvene for round two of the cocktail class afterward.
So just to warn you what the format is, so while we clear the table and get ready for the next cocktails, I'm going to do my, I had a couple of slides there. Dwayne, can anybody forward the slides? Oh, yeah. You may not be able to see them, but this is my trip down memory lane. So keep in mind, so this is the time is 1984, and so on the left, you can see we both had the same hairdresser. As I say, both fashion trendsetters, and then the other picture on the right is just a few months ago when I was down in Washington and got to have dinner with Frances and Diane. So we've known each other an awful long time. And when we were thinking about the program tonight, Diane brought up the idea, well, why don't you interview Frances? So if you go forward one more, Dwayne. So Frances has done so many things. One of the smartest ones was marrying Diane. So she doesn't get nearly the recognition. She's kind of hides in the shadows to some extent. She's very gregarious, talk to anybody, so on and so forth. But you won't know talking to her that she is a legend in the genetic counseling field. So she started the program at the University of Michigan, the first human genetics department in the country, and she's done just absolutely fabulous things. So these are two powerhouses that got together and have made such an impact in medicine and genetics in particular. So, so the last slide is my attempt at being clever. So I'm going to interview Frances, ask him some questions, and he knows I don't like surprises, so I'm not going to hit him with anything embarrassing or anything like that. But I think some questions for you all to get to know more about what he's all about, because he's done some, I don't know how to say, just spectacular things. The man has met and made friends with so many notable people around the globe. So you go into their house and there's a scarf in his front room from the Dalai Lama, right? He and Diana spent time with, is it Winona Judd? No.
Naomi, but
Naomi Judd. Yeah. So, oh my gosh, I'm blanking out now on the names. But so many notable people in the world. So my first question for you, Dr. Collins, is it would be ridiculous, I think, to ask you a question of what do you think is your most notable accomplishment, but rather name a few or tell us about a few that you feel the most satisfaction from, because you got such a list. What are the ones that really stand out you feel where you've had impact that gives you immense satisfaction? And we've already talked about Diane.
I was going to start there. Well, first, let me say it's really a joy to be here in Cleveland with all of you and with Mitch and this wonderful institute that we're here to celebrate on its 10th anniversary, and all of the good things that have happened so far and many more to come. So when Mitch suggested this might be a weekend not to miss, Diane and I were immediately bought into that as something we just want to be part of it, especially when it came to, making cocktails. I mean, gosh, how many times do I get to a meeting like this? Probably never. So I also have to tell a couple stories. I'm sorry, I'm gonna break with the plan. You already lost control of the fireside chat. Because, Mitch was my first graduate student, but he was much more than just a student in the lab. He also set the tone for the whole lab as the first person to get there. One of the most open-armed, warm, wonderfully encouraging, I've got time for everybody kind of guy. And that is true to this day. And I think that's one of the reasons Case is so fortunate to have him, is because of the way in which Mitch reaches out to everybody, gives of himself, and tries to make every scientist and every physician have a better chance at success because he's going to help them out. And that first project he got put on was just a very simple one. Can you find the cause of cystic fibrosis, which has vexed everybody for decades, and maybe you, a graduate student, can somehow come up with a solution? And it was a tough road. And anybody who had more impatience would have had a hard time sticking with it, because this is a 5-year slog through so many blind alleys and frustrations where you thought you might have something and then it always fell apart. Because this was back in the day where we had almost no knowledge about our own DNA instruction book. There are a few little islands here and there in a sea of ignorance, and you had to somehow sift through that and try to find what might be as simple as one letter misspelled. It turned out to be 3 out of 3 billion. Most people in the field thought this is probably never going to work. But this guy was absolutely determined to just bring every bit of energy and skill and determination to see if we could take some technologies that were sort of vaguely successful, just sort of being invented, and see if we could land on this gene. And ultimately, in 1989, that did in fact happen. And I'm sure we can talk more about that. I'm particularly thinking about CF because I came today from New York at the annual Lasker Awards. This is the US version of the Nobel Prize. And the Lasker Clinical Award went to 3 investigators who led to the success in the drug Trikafta that has now been a miracle for people with cystic fibrosis. So the whole American medical community decided this was the clinical achievement that was most worthy of acknowledgement here in 2025. And that was pretty exciting to see that happen. After all these decades of hard work, we can celebrate that. But I got to tell one other story about Mitch, which is not only was he a great guy in the lab, he also had a particularly important role in my personal life. So, Mitch, do you remember sort of the fall of 1992? Mitch by then has gotten his PhD. He's a postdoc in the lab, getting ready to figure out where to land next, which happened to be here at Case after a little bit longer. But he's also the person in the lab who has the most experience. He's the person everybody goes to saying, Mitch, can you help me make this experiment work? Because I don't know why it didn't. And I'm at that point single, not because I had expected to be. So I was single again. And I'm pretty miserable. And I'm trying to figure out whether there could ever be a chance for me to be with somebody of the opposite sex, or whether I'd better just plan to be alone forever. But I kind of was interested in this genetic counselor who actually ran the program at Michigan, but I didn't know whether there was any chance that she might potentially be interested in my asking her out. And I was terrified of that because I hadn't really done that since junior high. But I knew that Mitch and this genetic counselor named Diane Baker were friends, because Diane is friends with everybody and Mitch is friends with everybody, so they naturally are friends with each other. Do you remember being asked by me about whether Diane might potentially be interested in a conversation with me of something other than DNA?
Well, what I remember was actually Diane coming into my office, shutting the door, and say, I think I was getting signals. I'm paraphrasing, but that was the that was it. So then let me defend myself.
Mitch is totally out of control now. And it's going to be someone else in the audience who picks this microphone up next. So I decided, wow, Francis is a really nice guy, but I don't know if he's dating anyone. So I'm going to ask Mitch. So I wait till the end of the day. I go into his office. It's a suite of offices in an open area, and various offices off it, Mitch's being one. His door is always open. I walk into his office at the end of the day, and I close the door, and Mitch goes, because you never close the door to his office. I hopped up on a file cabinet, and I said, Mitch, I'm just wondering, do you know if Francis is dating anyone? And Mitch goes, Diane, we don't talk about those things. Fortunately, that night, Mary calls me. She says, I understand. There's a conversation going on. Let's figure out how to move this forward. So thank you, Mary.
All right. Well, my next statement is, there goes my...
Okay, you did ask a question. I suppose I should respect that since I was supposed to be your guest here. What things have accomplished besides your matchmaking of Diane and me, which led to this joyful marriage that we've now had for some 27 years. It is hard, Mitch, to sort of pick out in the course of, for me, what has been more than 40 years of running a research lab of various sorts. Even when I was directing the NIH, I kept my lab going because it felt like a good thing to do, keep me anchored in the reality of what science is all about and not just read about it. I guess I would certainly pinpoint that discovery of the cause of cystic fibrosis as very near, if not at the top of that list in 1989, because it was such a moment of light shining in the darkness about a disease we desperately needed to find better answers for. I'd taken care of kids with cystic fibrosis, and a few had made it to adults, and it was a really hard thing to say, I'm sorry, we just don't really understand this disease, and we don't know what to suggest as far as any advances. And finding the cause, those 3 letters, the famous Delta F508 mutation, was a start, but it was a start of a very long haul. And to see that now, in the point where anybody who has that particular 3 base pair deletion, and a bunch of others as well, now having a drug that is dramatically successful, Trikafta, and many of those individuals who didn't expect they were going to have much of a potential life expectancy or even talking about retirement, which is just a dream come true. So how could one not sort of mention that as a high point? But running the Human Genome Project, being in the place of managing an incredible international team, 6 countries, 2,400 scientists all working together for the same goal, at a time where in the beginning of the project, most scientists were opposed to it, thinking that it would never work. We didn't have the technologies to read out 3 billion letters of anything, and it was probably going to waste a lot of money and end up as a failure. But everybody pulled together, and they believed in the goal, and they stopped worrying about who was going to get the credit, and everybody made the job happen bit by bit. And we finished 2 years ahead of schedule, $400 million under budget, and started something that continues to this day as an absolutely revolutionary way to understand how life works and how disease happens. And to see that now in its only partial flowering, there's much more yet to come, is just dizzying and wonderful. But much credit again. I happen to be the field general who sort of had to marshal the troops to try to make it happen. But it was those 2,400 scientists who did the work and achieved that. I guess as NIH director for 12 years, I managed to start a couple of big projects that I'm hoping are going to make a big difference. They're still works in progress. One was the Brain Initiative, to really try to figure out how those 86 billion neurons between your ears do what they do, and how sometimes that goes awry in disorders like schizophrenia or PTSD or autism, things that we just desperately need to know more about. And we don't have the basic understanding of those circuits to know how to understand when things go wrong. And that's now 10 years along, and our understanding of the human brain is just completely gone through a total revision, and many more to come, because now we have data. And we recruit a lot of engineers, by the way, to help us figure this out, because that was the kind of attitude we needed. And then this other project, which also got started about that same time, to try to understand precision health, how it is that we're all different. We have different environmental exposures, we have different genetic endowments, we have health behaviors that vary. How does all that fit together in terms of whether we are healthy or whether we're starting to get sick? And how could we use that information for better prevention and better management when illness happens? We desperately want to know that. One size fits all is not going to be the answer. But if you really want to individualize this, you have to have a huge database of individuals who are willing to share everything about their health, including their genome sequences and their medical records and all kinds of lab tests. And we now have 880,000 Americans who are doing that. If you're not part of them, it's not too late. We're going for 1 million. Joinallofus.org is a fashion where you can become part of that team. And what we're learning already is pretty astounding. And there's some 12,000 scientists now who are authorized to have access to the anonymized data sets and are making inferences every day about things that previously we just weren't quite sure about. I guess that's a long enough list anyway.
Well, no, that's great. And so I will make we are after we're done right here, we're going to go back and make some more cocktails, but then we're going to open it up so you all can ask questions. And I know, where's Jim Gardner? So Jim's going to ask you, I'm going to tell you so you can prepare for the answer, is what is standing in the way of using the personalized health information? So, right now, still the docs are going through their old ancient rubrics of right? So
Yeah.
we've
Yeah.
got all this information. So what's standing in the way?
A lot of it is just the unfortunate inertia of the translational process from when you have a research result that you know is going to help people and actually getting it implemented as part of the clinical practice of medicine. That interval, which you would think ought to be like, a few months, when people actually measure it, it's more like 20 years. And there are all kinds of factors to that. A big part of it is just reimbursement. I think you can make an extremely strong case right now that every one of us would benefit from having our human genome sequenced carefully, accurately, placed with appropriate privacy protection in our medical records, available if you happen to need to know whether this drug you're about to be prescribed is going to be a good fit for you or not, this whole area of pharmacogenetics. But most of us have not had that experience. And why is it? Because third parties won't pay for it. So much of our healthcare system is driven by that question. Will the third party pay for it? And to get that agreement just takes mountains of data over a decade to actually prove that this changes outcomes, when it very obviously will. But you want CMS and the third-party payers want to see more than that. I mean, that might have made sense when it cost, $100,000 for a genome sequence. We're getting pretty close to $100 now. That's so much less than a scan. And you only have to do it once. Why are we not doing this? It's because of our crazy healthcare system that won't endorse things until everybody knows they've been working for years. That's part of the problem. The other part of the problem is we don't have all the data we need. That's why this All of Us program with those 880,000 people is so valuable, because we could begin to actually figure out what kind of combination of genes and environment is something that you really want to think carefully about. Should that be an individual who changes their health behavior to try to reduce the risk of some kind of outcome? Really empower people to give them that kind of information about how to stay healthy, how to have a long health span and a lifespan, but a health span's really good too, as opposed to right now where a lot of us just get, here's this laundry list of the things you should do. And most people end up ignoring it because it doesn't really sound like it's about them. Well, it could be about them. And we've kind of known that for 20 years now, since the genome sequence was first put in place and you could start to understand where this would lead you. But we are so slow to implement it. The UK is doing a little better than we are, but they're not doing that great either. There's so much molasses in the system that needs a good hard kick.
Yeah, so you may not be surprised that we think a lot alike on these things. Whether that's the reason we're such good friends, I don't know, or if it's a mentor-mentee kind of thing. But
or a mentee-mentor thing. It goes both ways, Mitch.
But having, when you go in and get your blood pressure taken, that's what it was that day. When you get your genome sequence, that's what it is your entire life. So you can go back to that information again and again and again as things happen. And we're just not using it to the extent we could. And so hopefully we'll get there. But this is our public service announcement that we really, really need to push our government officials to keep supporting science. So, I wasn't going to get on the soapbox on this, but, this is an area that used to have incredibly strong bipartisan support and now has been politicized. And so we've got to get back and convince them that, no, your health care doesn't matter if you're a Republican, Democrat, Independent, or from Mars. It's the same for all of us. And so we've got to convince them that this is something that shouldn't be controversial. So.
disease does not care about your party affiliation or whether you're black, white, or brown, or whether you're rich or poor. Disease also doesn't care if you happen to be young. What you're doing here, focusing particularly on children's illnesses, is so critical because those are kids with their whole life in front of them, and yet with something that comes along totally unexpected by them or their parents, we desperately need better answers there. And we're getting there in a pretty exciting way, as we can talk about that. I've never imagined the kind of progress that's happening in terms of approaching rare genetic diseases, most of which affect children, as what we can do right now. And yet, just picking up on what you said a minute ago, we have this strange paradox of exponential opportunity in expanding our ability to make those discoveries and save lives, and an unprecedented cut in the support for that in terms of NIH. And that just seems so odd. We can show that support for medical research by the federal government has been absolutely critical for most of the successes that we could talk about. Cystic fibrosis. Mitch, if you and I had not had that NIH grant and support from the Cystic Fibrosis Foundation back in the 1980s, we couldn't have worked on that problem. And that support went on for another 10 years with not a whole lot to show for it, but trying gene therapy, trying drug therapy, wonder something's got to work here, until eventually it looked like, oh, maybe there's a clue here. This is starting to work a little bit, and it's time to do that transition into the private sector, to Aurora and then to Vertex. And then we end up with a success story. Most people don't know that. They don't know that how this miraculous cure for this terrible disease came about depended on those 33 years of this ecosystem involving the federal government through NIH, involving philanthropy through the CF Foundation. I'm glad we have somebody here who's a big part of that. And also the biotech industry stepping in a rare disease, which was pretty risky at the time because nobody thought it was going to work. And most importantly, the patients who said, okay, I will be willing to sign up for a clinical trial without any confidence that whatever's being proposed is actually going to work. But we never know until you do that trial. And that's such an important part of this amazing set of advances that have happened. It's all because of patients who have had their trust to take part in things that were promising but not at all certain. That's the kind of thing I know you're trying to do here, is to create more and more opportunities like that. They won't all work. We have to expect that. Medical research is really hard. Failure is all too frequent in each of our lives, even when you really want something to work, but you got to be rigorous about it. And the only way to be rigorous is to run those clinical trials that we ask people to take part in, knowing that that may or may not benefit them, but we'll learn something from that that may ultimately benefit somebody else. That's what you're doing here.
And this is the second plug to ask you if you really want to come into the lab and see what we're doing. Because stuff we would not have thought was going to happen in our lifetime, the ability to actually go in and fix a gene that causes the disease, at least I didn't think we were going to be able to do that. And then all of a sudden, folks come up, Jennifer Doudna comes up and says, hey, we've got this thing called CRISPR. It'll go in and change DNA any way we want to. And so we'll show you in the lab, say, look, we can fix the CF gene. We can fix this gene, that gene. We're now at a point, we're so close to, I won't call it the finish line, but making a clinical difference where we can do it. We now need to work on making it work well enough. How do we get enough cells fixed and the right cells fixed that we can do this? So I'm sure you feel the same way, that we didn't think it was going to happen. Yeah.
How many of you saw the press reports maybe 6 months ago about baby KJ? Okay, let me tell you about baby KJ. This is such an encouraging story because it's a glimpse of where we could go on a much more scaled up way. So baby KJ was born in Philadelphia. No indication there was any problem. But within the first day, an alert nurse noticed something isn't quite right. The baby seems a little jerky, a little bit not in a normal tone. So quick workup was done baby KJ has ammonia levels that are off the charts. That is a very bad situation for anybody who studies genetics, because that means baby KJ probably has a genetic mutation in one of the genes involved in the urea cycle, which is essential to keep your body okay. And sure enough, they quickly, very quickly, because you can do this now, because all the technology from the Genome Project sequenced all the genes that might be involved, and sure enough, this baby was missing a functional copy, two functional copies. It's a recessive disease of a gene called carbamyl phosphate synthetase. And that, without treatment, is lethal. Not immediately, because you can kind of tide these kids over for a few months, but few individuals of this have ever lived past about a year and a half. Well, because of all the progress that has been made in developing ways to do this with gene therapy approaches, Kiran Mosanuru, who was a pediatrician who had worked in the private sector for a few years and now was back at CHOP, said, let's just see what we can do. Let's see how fast we can develop what you would call a bespoke gene therapy just for KJ that might fix that mutation. The target is the liver, because the liver is the thing that does the enzymes that are necessary for this urea cycle to work. And you have to come up with a way to then deliver a gene editor, a CRISPR kind of second generation effort to go and fix that mutation in that gene very specifically. Don't want to make a lot of mess of the rest of the genome. This is like doing find and replace in your word processor. When it works well, you say, okay, I want to find that word and I want to replace it with this word.
This is not autocorrect.
No, thank you. We're trying to avoid autocorrect for the genome, but find and replace. And in that space, they developed the gene editor that had that targeting ability. They applied a particular vector that would get it to the liver. They ran through animal experiments. They quickly made an animal model of that same mutation, ran animal experiments, convinced the FDA that this was worth trying, especially given the potential lethality. And 6 months later, this baby received the first dose and another dose 2 months after that. I recently heard from people who are following him that KJ, now a little over a year old, appears to be normal. Unbelievable achievement. But it's not just unbelievable for this N equals 1. It's also because it was possible to do this in a space of just a few months to come up with a treatment that worked. Now, it cost $1 gazillion to do this, and that's the problem. How do you scale this up for all these rare diseases that may affect only a few individuals in the world? We have to figure that out. But that's achievable. That's kind of a manufacturing engineering problem. If you can do it, and the regulators have to be okay with going along with not doing everything like you would for a blockbuster drug that's going to be used for 2 million people. You got to have shortcuts here when a kid's life is at risk. And you have to accept the fact that that means the benefits and risks are different in that situation. But I'm so inspired by that. My own lab is working on another one of these rare diseases. How many of you have heard about progeria? So you've probably seen pictures of these kids who are aging at 7 times the normal rate. Their brains are developing like normal children. Their bodies are turning into little old people. And without interventions, they will die at age 13 or 14 of heart attacks or strokes. Losing all their hair, their skin looks all wrinkled up. They look like somehow they're 90 years old when they're 13. We discovered the cause of this now some 20 years ago, and it's remarkably reproducible. Almost every child with this disorder has a single misspelled letter in a gene called lamin A. What should have been a C is a T. That's all it takes. Well, if you're going to try to design an approach to this advance to do the find and replace, the CRISPR gene editing, seems like the right strategy. And so working with the real guru of gene editing, Professor David Liu at the Broad Institute of Harvard and MIT, now for 7 years, we've been trying to see, could we actually make this work and help prolong in a happy way the survival of these kids who are so tragic otherwise. And they're so courageous, they're so precocious, they're so determined to make the most of the little time they have. And again, their intellectual development is stunning. So we built a mouse model of this with the human version of the mutation, a humanized mouse, as you call it, which is a critical thing to be able to do. Let nobody tell you that we don't need animals anymore. We really do for something like this. And with David's help, we designed a gene editor, and we infused the mice that had progeria with this, who normally only live about 4 or 5 months, and some of them live for 2.5 years. It was astoundingly successful. And now we're in the process of going through all of the other steps you have to go through to convince FDA that it's okay to do this for a human trial. And sometime in the next 12 months, we will treat the first progeria patient. Fingers crossed. All kinds of prayers that this will actually work in the human the way it did with the mouse, because there's never an assurance of that until you try it. But it's just one more example. But yeah, to take this on, though, in the bigger scale, there are, depending on who you talk to, 7,000 to 10,000 rare genetic diseases like this. Many of them ones you study here in this remarkable facility, this institute. And we know the specific misspelling now in most of those, because diagnostics gotten really good because of genome sequencing. If we could figure out how to do this in a fashion that's scalable so that every one of those could be approached in sort of a standard way, the right gene editor, the right vector to deliver it to the right tissues, we could go after all of those and not take the next 3,000 years to get there. I think that's doable. That makes me really excited about where we are. But it's going to be costly and it's going to be expensive. It's going to be, at times, failure prone. We're going to have experiences where it didn't work and we thought it would. And it's going to take all the energy and all the creativity and all the determination of a place like RICH to make those kinds of things actually happen, because it takes those kinds of committed, really smart investigators to push this agenda forward. That's what I think is, in my view, one of the most exciting things that's happening in all of medicine.
Yes. And if you've come through the lab, you'll know I'm telling you the truth, that you meet with the people, they are so excited about what they're doing, because we can see it's tangible. It's not science fiction. It's doable. And we just have to get over some of those, social, economic hurdles to make it happen. Just one more example, and I think it looks like stuff may be ready for the next. But please, if you take no other message home, tell your friends, because we know the numbers are such that every single family is affected by a genetic disorder of some sort. It may not be you or your child, but it will be your aunt, your uncle, your niece, your nephew. Somebody will. So it's not hard for us to convince people why this is important. We need to convince them about how doable this is if we get the support. So, all right. So why don't we'll reconvene, and I'll ask my questions then. So,
And I won't answer them probably.
All right, as we get kind of settled back in here, everybody, who's inspired? I am, right? I'm really inspired by that. Okay, round two. We all ready? All right, as we established in round one, back up, please. Not yet. This one right here. Nope, you're going the wrong way. Okay, everybody. So as we learned in round one, Tiki peaked and went down and came back up again. So we are going to talk about the coming back up again. When Tiki came back, modern mixologists loved the classics, but they also didn't want to rest on their laurels, right? So they decided that they would tweak, in some cases, old recipes, and in other cases, come up with brand new ones that no one had ever seen before. We are going to do a little bit of both of that in our second round here. And we're going to take advantage of something called the Mr. Potato Head approach to mixology. Who remembers Mr. Potato Head from your childhoods, right? So you had your potato and you had your pieces and you could customize it however you wanted. And Mr. Potato Head might have had a mustache or not. He might have had a hat or not. He might have had a smile or a frown. He might have even had a wife, Mrs. Potato Head. We are going to do a Mr. Potato Head approach to mixology. So what mixologists do is keep the base. In this case, the base is rum, right? But they swap out different things. And by swapping out ingredients, just like if you were cooking and you swapped out a few ingredients, you got a different dish. If you are making a cocktail and you swap different ingredients and you know what you're doing, you get a drink that has a different flavor profile. We are going to make a drink tonight called the Jungle Bird, which is a classic tiki cocktail. I'll tell you more about it in a minute. And another tiki drink, a modern tiki drink called the Kingston Sound System. And I'll tell you about that one as well. Two ingredients are different in this drink compared to that drink. And they make all the difference in the world. So, Mr. Potato Head, keep that in your brain. And let's move on to talk a little bit about rum, our base, our potato, right? Rum, we could spend a whole class talking about and still not cover the subject. Rum is an amazingly varied, complex, interesting drink with a fascinating history. We'll talk a little bit about that in a second. You can see a few of the many different kinds of rum up here on the table. Rum, at its heart, is a product of sugarcane. So it's either the distillate fermented of sugarcane juice or molasses. And molasses is actually kind of the industrial waste, if you will, of sugar making. Rum has a fascinating history. 1600s in the Caribbean. Sugarcane grows naturally, right? Very rich Europeans and Americans in the later 17th century and 18th century established sugar plantations, primarily in the Caribbean. And they're growing sugarcane to make sugar, which is a incredibly valuable crop. So, not content to make their money from sugar, these sugar barons with their sugar plantations decide they want to double dip. They want to come up with something else besides sugar. So, they figure out, with some help from native populations in those areas, that you can extract the juice or take the molasses that result from sugar making, and you can turn that into rum. Where does rum come from, the name rum? Anybody know? Well, the answer is nobody really knows for sure. There are a few theories. Sugar, the Latin word, is sacrum, right? It might be an abbreviation of that Latin word sacrum. There's an Old English slang term called rumbustion. It means chaos. And you could imagine that someone who drank a lot of rum, which a lot of people did in those days, might cause a lot of chaos. So maybe rumbustion is the origin of the word rum. We just don't know. But these sugar barons on their sugar plantations figure out how to make rum, but they've got to get it back to Europe and to America, which is being colonized at that point, because that's where the drinkers are, right? So the only way to get it back is on ships. And what happens on ships? You have sailors on ships, and in those days you had pirates that loved to pirate what was on these trading ships. The sailors loved rum. They loved rum to the point where the captains of the ships had to do something to control the drinking. So the Royal Navy, the British Royal Navy, actually started giving sailors a ration of rum. And that's a practice that actually continued until 1970. As late as 1970, British sailors were still getting their daily ration of rum. Pirates, of course, would attack these ships and take the barrels of rum. So, the whole yo-ho-ho and a barrel of rum, well, that's where that came from. So we have this trade of rum to Europe and the colonies, became very popular. As time went on, of course, rum by itself wasn't enough, and people decided they wanted to start making cocktails from it. So that's kind of where we jump off into tiki cocktails. Remember, Don the Beachcomber sailed the Caribbean, sailed the South Pacific, knew a lot about rum from his travels with his grandfather, even though he was probably underage. But he developed a taste for rum, and he came up with these marvelous, inventive tiki cocktails. And that practice has continued until today. Next, Nurse Andrea. There we are. So that leads us to the Jungle Bird. That is our classic tiki cocktail. Where did the Jungle Bird originate? So in 1957, a guy named Jeffrey Ong, this guy right here, was the bartender at this place right here, which is the Kuala Lumpur Hilton, which had a bar called the Aviary. And Jeffrey Ong decided he wanted to create a drink on that bird theme, aviary. So he created the Jungle Bird. Jeffrey was an inventive sort. And, go ahead, next. He decided he wanted to include an unconventional ingredient. So who knows Campari? You guys know Campari? All right. Campari is an Italian liqueur, a digestive, an aperitif. And you probably most know about it in the Negroni, a classic and Italian cocktail. You don't think about it in tiki much, but Jeffrey Ong did. Campari, what do you notice about it the most? That color, right? This distinctive scarlet color. Where do you think that comes from? Well, today it does, but back in the day, back in the late 1800s when Campari came to be, there was a bug called the cochineal, which is this distinctive reddish brown color. And it lives in Central and South America, where it hangs out on cacti and lives its little life. And in order to ward off predators that like to eat cochineal, it has a natural defense. It's called carminic acid. Carminic acid is bitter, and other predators of cochineal don't like the taste. So that's its natural defense. Someone somewhere figured out that if you took these little bugs and you dried them and you crushed them up and you extracted the carminic acid, you could create a coloring called carmine. Carmine is a natural dye, and you would find it not just in the original Campari, but in things like lipsticks or clothing dyes. It was all over the place. Modern Campari does not have carminic acid, carmine dye in it. It has man-made food colorings in it. Except there are some people that are kind of on a back to original kick, and you can find some distilleries that do make Campari the original way. People are shaking their heads. Not for you, right? Okay. So Jeffrey Ong says, I want to try and make the Jungle Bird with Campari. We'll talk a little bit more about that in a second. Shannon Mustafer. Shannon Mustafer is an inventive bartender. Shannon worked at the Gladys Caribbean Cafe in Brooklyn, a wonderfully inventive bartender. And Shannon decided that she wanted to create a riff on the Jungle Bird. She was inspired by reggae, a reggae song called Skylarking. And she kept the rum, although she used a different kind of rum. And we'll talk about the two different kinds of rum here in a second. she substituted a different kind of rum. Instead of Campari, she used something different. And instead of, don't go yet, back up. There you go. We'll talk about what else she swapped out in a second. I want to mention about the two different kinds of rums, the one in the Jungle Bird and the one in the Kingston Sound System. So in the Jungle Bird, we are using this lovely stuff. It's Gosling's Black Seal rum. And you can see it's this beautiful, dark coffee color, right? If you want to take the lid, those of you, I'm sorry, I forgot to say, half of you are going to be making Jungle Bird, half of you are going to be making Kingston Sound System. And then if you want, we have straws, you can sample each other's drink and you can see how different just these slight swaps are going to make your drinks, right? So Gosling's Black Seal rum, if you have that on your placemat and you want to take the lid off and smell it, rum has this real unique characteristic. It's called funk. Rum aficionados talk about the funk of rum. Another word that sort of means the same thing is hogo, H-O-G-O. Hogo comes from the French phrase au goût, which means high taste. Both of them are attempting to describe this essence of rum, okay? Different rums have different tastes, but they all kind of have this funky property. It can be kind of grassy, kind of vegetal, kind of evocative. But if you smell Gosling's, you get a little butterscotch and a little caramel and a little vanilla. Anybody pick that up? Anybody pick up anything else? So that's Gosling's Black Seal rum. That's what's in Jungle Bird. In Shannon Mustafer's Kingston Sound System is this big boy right here. This is Plantaray OFTD. OFTD stands for Old Fashioned Traditional Dark. So it's a dark rum. Rum is characterized often, there are lots of different ways of talking about rum, but one way of characterizing rum is the color. So there's clear rum like Bacardi that you might know, and that's clear because it's been filtered. There are darker rums, and the reason there are darker is because they've been aged in oak barrels like you do with bourbon. And when you move the alcohol in and out of the wood in the barrel, it picks up tannins, which give it color and flavor, sweetness. So rum does that just like bourbon does. This is an overproof rum. You can imagine what overproof means, right? It's high proof. It's high octane. This is 138 proof. It is a strong, strong rum. We are not putting as much of this in our drinks in this as we are in Jungle Bird. But if you take the lid off, if you have Plantaray OFTD and smell it, it's got a little bit of a different smell. It's spicy for sure because it's a high proof rum. If you want to give it a little sip, you're welcome to do that. You'll have enough still to make your drinks. Okay, so ingredient swaps. Next slide, please. Kingston Sound System has this lovely, pretty rare bottle called Suze in it. Suze is swapped out for Campari. So no Campari, no red. It's a very yellow drink as opposed to the red color of Jungle Bird. Suze is a French liqueur, also an aperitif. It's made from the bitter root of a beautiful flower which grows in the Alpine mountains of France. This is called gentian. So this is a gentian liqueur. It has a very distinct flavor, color, and taste. You can see it's very vivid yellow. If you were to drink it straight, and it is a polarizing drink, people like it or don't like it, but it tastes, it's got a tartness like grapefruit, it's kind of lemony, it's got a floral taste to it. It smells kind of vegetally, if you can take a snip. I wouldn't recommend drinking this by yourself. You would drink it and go, wow, I don't want to drink anything with this in it. But trust me, it's going to be okay. The other ingredient that Shannon Musfer swapped out, instead of the pineapple, she went with something called soursop. Anybody ever heard of soursop? So soursop is this. Let's pass it around. Soursop is the fruit of a flowering evergreen tree that grows in Central and South America. If you were to cut that open, you would find this kind of white coconut-looking kind of meat to it. It's got a softer sweetness than pineapple juice does. Soursop is incredibly expensive. That one particular one right there cost $11. If you were to buy a case, which is how it's sold usually, $350 a case. You can only get it in certain months of the year. I found that at the Westside Market last week. I was there earlier in the summer and couldn't find it, but fortunately it was back in stock. The good thing for us, if you want to replicate this drink, is that you don't have to go buy soursop fruit. It's so messy to try and extract the juice from that soursop. First thing, it's hard, right? And the pulp, the meat of it, is really messy. You would want to do it in a bathtub without any clothes on, trust me. But luckily for us, Goya, which makes a lot of ethnic foods, has extracted the nectar from soursop. And you can find this in a lot of grocery stores, particularly on the west side. So we have soursop nectar. We are also in both of these drinks using agave nectar. Agave nectar is made from agave, of course. Why are we using that? So it adds a sweetness which both of these drinks need. Agave mixes with your drink better than sugar, crystal sugar would, and better than honey would. Some cocktails have sugar, some cocktails have honey. Agave nectar it just is easier to mix, and it doesn't add a real distinct flavor, and we don't want to really detract from the other ingredients that we have here. So we're going to make these two cocktails. Take your rocks glass, which each of you should have, and put that where you can find it, and take your shaker tins, which we've rinsed out for you, okay? And in your big shaking tin, put your ingredients. Doesn't matter what order, add them however you want. Okay, and the ice is coming around, so we're going to put our ice in the small one. We're going to fill it up about the same level, about 3/4 up, right? Everybody got their ingredients in the big one? Yeah. I'm going to give you a second to add your ice and finish up with your ingredients. All right, and we are also, our lovely assistants are coming around with big ice cubes. These go in your rocks glass. These big square cubes, put them in your rocks glass. Just one per glass. All right, everybody got your shaker loaded up, right? Okay, you got your rocks in your rocks glass. Take your shaker tins. Remember, we're going to take the ice and pour it in and mate the shaker together like we did before at an angle, nice angle, right? Give it a tap to give it a seal. Hold it on the bottom. Hold it on the side. Get a good grip. Don't wear your cocktail. Give it a shake. 20 seconds. Come on, I want to hear you. Workout. Come on, come on. Come on, let's break a sweat. Let's go. Shake, 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 shake. Come on. Oh, you sound good. You sound great. All right. All right. Shakers down. Give it a little squeeze to break the seal. Give it a tap to get all the good stuff out of the top. Put your Hawthorne strainer on. Everybody get it unsealed? Can you get it? Here, give it a squeeze on the bottom right there. Squeeze the yep. I can get it. There you go. Everybody pour their drink. All right, don't drink yet. Don't drink yet. We got to make the garnish. Tiki is nothing without garnish, right? Okay, so on your table, if you're a jungle bird, you have something that looks like this. We're going to make a jungle bird. It's only going to take 2 seconds. Take your toothpick. Jungle bird, folks. Toothpick. Poke your Luxardo cherry, right? You got it on your toothpick. Take your pineapple slice and poke your toothpick through the cherry, through the pineapple, all the way out to the other side, right? And then take your pineapple frond. One here, like that. One here, like that. Voila, jungle bird. Put it on your glass, just like that, right? Okay, Kingston sound system, folks. You got your lime wheel. Take your lime wheel. It should have a notch in it. Just put it on the edge of your glass. Take your pineapple frond. You can put it right in the glass like that. Yours is not quite as elaborate, but it's nice, right? Okay, everybody got their garnish in place? Let's give these a try. Tell me what you think. Now, these are going to be a lot less sweet than the first time around. They're going to be a different flavor. They're going to be a little bit bitter. You might like it, you might not, but it's an experience, right? That's what we're here for, is an experience. If it's too bitter for you, we have agave nectar, and you can squeeze more in. Where's my agave nectar assistants? Anybody need extra agave nectar? Anybody? Where did it go? Where did it go? Okay, while we are searching for the agave nectar, I see a bunch of sort of strange faces out there. I can't tell whether you like it or not. These are different drinks. They are more advanced drinks. They are not for everybody, but they're an example of what tiki can be. And what I hope you take away, if you take away nothing else besides your glasses, which you're going to take, and your recipe cards and your shakers, is that when you go home and you set up your home bar, and I know all of you are going to do that, experiment. Try things. Taste things. Swap out things. Experiment and be creative. That's what this is all about, is to have fun, be creative, and enjoy all of our company. Last slide. If you want to learn more about anything that we talked about tonight, here are 4 good books. Rum, A History of the Drink. It's a lovely book, and it tells the story of rum through a bunch of different cocktails. Smuggler's Cove is a great kind of modern tiki cocktail book. Shannon Mustafer of Kingston Sound System, this is her cocktail book, her tiki book. And Tropical Standard by a wonderful guy called Garrett Richard, who has a wonderful tiki bar. If you're ever in Brooklyn, Sunken Harbor is the place to go. Garrett Richard's bar. Highly recommend it. Any questions? Any questions about rum, tiki, cocktail, whatever? Don't have to have one, but happy to answer them if you have them. Well, that's what I've got for you tonight, folks. And we are now going to thank you all. Thank you all. And I believe that we might have some musical entertainment coming. So enjoy your drinks and let's listen.
So next step, so we're going to have question and answer for Dr. Collins, but he also has a musical interlude here for us as well. So he's written some lyrics for, I don't know what the song is, but I think it's a well-known one. Is that fair? No. No? Okay. Well, he's written new lyrics for it anyway.
Now?
So we can do the question and answer right now if okay.
We should keep them in suspense a little bit.
Okay.
Let's do the Q&A first.
All right. So I'm going to play Phil Donahue. You saw me do 60 Minutes. Now
I need
it's
my
Phil
drink.
Donahue time. So for you to ask questions, I'm going to start with the first one. So here's a guy who headed up the biggest medical research program in the world, not the country, the world, and but is a pretty modest person about it. You won't hear him bumping his chest about it, so I get to do it for him. So, but part of the amazing background here is here's somebody who was homeschooled for most of his school years, right?
Till 6th grade.
Till sixth grade in the hills of Virginia, right?
The hills, yes.
Not, yes, okay, mountains, but not an environment you would typically say, well, there's somebody who's going to go on and take over and lead NIH and things. So, so my question for you, Francis, is what do you think, looking back, what do you feel you missed out on, and what do you feel you would have gotten no other way?
That's a great question. I'm the youngest of 4 boys. My parents were doing the 60s thing long before it was the 60s. My dad was a college professor. My mother was a playwright. But they bought a 98-acre farm in the Shenandoah Valley and tried to be farmers on top of that with no mechanized equipment. So it was all done with workhorses, and it was hard work. And they thought that their 4 boys would do better being educated by them because of their skills in getting people excited about learning. So my oldest brother didn't go to any kind of formal schooling until college. As the youngest, I think my mom kind of ran out of energy when I got to grade 6. And so I had the best of all worlds. The first 5 grades being taught by my mother and father. And again, people with a great deal of excitement about learning things. It was mostly in the area of music and theater and literature and history. And my mother was a pretty good mathematician. There wasn't any science in those first 5 grades because that isn't what they did. But when I got to public school, I had the chance to get excited about science from a very good teacher in chemistry. So what I learned that was special was the love of learning. My mother could create that joy in discovering something for the first time that I still hang on to and long for, and that's what science is all about. And she could also make it clear that education wasn't supposed to be boring. It was supposed to be exciting. It was supposed to be fun. And we had no lesson plans during what I can recall as my first 5 grades. It was, what's interesting today? Let's pursue that. Let's see how far we can take it. And then when it stops being interesting, let's do something else. That was a gift. So I learned to love learning. What I didn't learn was how to be sociable, because growing up on this farm, my only real companion was the brother who was a year and a half older than me. And we alternately were friends and enemies. And I can't remember how many times my mother said, look, if you're going to kill yourselves, each other, please go outside and do it. I don't want the blood in the floor here. So it was a little strange when I finally did end up in a more social situation. I didn't know what to do. And I was also 2 years younger than everybody else in my class because my mom had taught us to move forward quickly. So let's just say my high school years were a little tough. The only refuge I had was to become a nerd and hang out with the other nerds. All in all, it was an incredible gift.
All right. Well, I know I could hit you with a button, even though I think I know you fairly well. I think I could go on forever. I want to make a couple other connections in here. So those of you who know me know my life is built on my network of people that I know. And I'll, just a couple. So Brenda Kimball here. Brenda's dad was my high school biology teacher. So there's one, right? It's just everybody knows everybody through a different mechanism. Tom and Mary Beth, who got this whole thing started, right? So our daughter Elizabeth and their son Dan, they were friends in high school. So that's how we got connected. Anna Mitchell here. So Anna and I were both graduate students in Francis's lab back just a few years ago. Yeah. So Anna now heads up the clinical genetics program at Rainbow or UH, and is just I don't know anybody who has recall the way she does. She doesn't forget anything, so be careful what you say tonight. But anyway, I could go around the room and tell you the stories about everybody and how we know each other, but that's been the magic about how this whole thing has worked. And everybody telling somebody else and so on and so forth. And it just brings people together, and we're able to do so much more by it. So I'll shut up now. And so who wants to ask the next question? And don't be shy. So Kim, or Jim, sorry, I saw a hand go up over there. Oh, well, see if you get the same answer twice.
No, it's okay. No, so just so you know, I'm a pharmacist by background, been in the industry 100 years, but I have the fortune of working for one of those ugly pharmacy benefit managers that's owned by a health plan. And we did a pilot in Michigan on pharmacogenomics. And that's why I asked Mitch, we had this discussion about this. But do you think a pharmacist has a play in this? Because what we can do is, what we did in the pilot was, we had pharmacists have discussions with the members, our patients, to talk about, if you did this test, then we'll know if these drugs are effective. And then we did the test, and what it was like, 89% of the patients that were taking drugs probably shouldn't be taking those drugs, probably needed an alternative in just a small sample set. But what do you think about the role of the pharmacist?
I think it's critical, and I wish we had gotten further with that. I love hearing about your pilot project because the way this works, everybody, is you have variations in the genes that are involved in metabolizing drugs. And that means you might be a fast metabolizer or a slow metabolizer. And that makes a big difference in whether the standard dose is right for you or whether it's too high or too low. And you'd really like to optimize that. Plus, you have variations in the receptor for that particular drug that might mean that drug's just not going to work for you because your receptor is shaped a little different. The drug's not going to stick to it. Or you might have a variation in the toxicity side of it, whereas most people are like, okay, I can handle that. And for you, this might be a really serious side effect, and you'd want to avoid that drug at all costs. There are 100 drugs in the FDA compendium that have on the label, there was information here about genetic variation that should be used, if possible, in deciding whether this is the right drug at the right dose for the right person. We're not using it. Because when the prescription is written, the genome sequence is not there. The doc doesn't know what to do with it anyway. The pharmacist could be in a great place to do this as an opportunity to sort of check before that first pill actually goes into the patient. Wait a minute, is this a really bad idea? I would love to see that done. But until we get to that point where the genome sequence is available for all of us, so you just look it up instead of having to send off a test and wait for 2 weeks, it's going to be really hard to implement it. I'm really glad to hear it's being worked on. Do it, man.
All right. Another connection here. So, Deb, the reason that Jim got into pharmacy is because of your family. So, yeah. So, it's all about people, right? All right. Yeah. Joy.
As you well know, we're at a moment in time in history where, that we're really losing the public on trust in science. And it's tragic and very, very concerning. And I think our health will be highly impacted by it. What messages might you advise us to give to the public to help them understand scientists are trustworthy, we're working hard, we don't always know the answers, but we're doing the best we can? What what might you advise, for example, a university like Case Western Reserve, as we try to tell the public what this is all about?
That is a great question. And I know you're a leader in the university, and this must be something that vexes you and all your colleagues every day. You can see the gradual trouble that we've gotten into in terms of distrust of science starting a ways back, but accelerating dramatically during COVID, which is such a heartbreaking statement to make. The development of the mRNA vaccines that were put forward by Moderna and Pfizer, but which began as research at NIH in our Vaccine Research Center on January 10th, 2020. I remember that all very well. Will be seen, I think, by science historians as probably the most significant advance in the history of humanity as far as science rising to the occasion to save lives. Estimates are 3.2 million Americans are alive now because of those vaccines. And of course, none of them know if they were among those 3.2. I don't know, maybe I'm one of them. Maybe I shouldn't be here without that vaccine. But along the way, with a whole lot of misinformation coming from social media, with some flaws in the public communication about COVID vaccines, and that some of that's on me because I was doing a lot of that communication also, people began to distrust the information as being reliable. I wish we had, every time we were giving a message from public health in the midst of this crisis, have said the following: we are still learning about this virus. We really don't know what I wish we did about how it's going to affect people. I'm going to tell you today the best information that the experts can come up with, but it might need to be revised. It probably will need to be revised. That's how science works. We learn new things. We come up with better answers. We didn't say that. And so when a recommendation was made in all good faith, trying to help people protect themselves and their families, and it turned out to be wrong a few months later, and it had to be changed, people began to question whether the experts are really experts at all, or whether they're manipulating people for some other purpose, which is truly tragic, because believe me, the public health people had no other secondary gain. We were trying to save lives. But along the way, and this was the saddest part of this whole story, the vaccine got developed in 11 months. No vaccine had ever been developed in less than 5 years. And its efficacy in that phase 3 trial that was done by Moderna, a separate one by Pfizer, involving over 30,000 people, showing 95% efficacy and no significant indications of side effects of a serious sort in those 40,000 people, although we later learned about rare difficulties with myocarditis, fortunately all reversible. So it was like the best outcome you could have imagined. I prayed about this every day in 2020. And the prayers seem to have come true. But for 50 million Americans, it was not convincing. It was not something they could believe in. And they said no. The Kaiser Family Foundation done a very careful analysis of what the consequences of that are. And they published an estimate that 230,000 Americans died unnecessarily between June of 21 and April of 22, just in 9 months, because the vaccines were fully available at that point, but a lot of people said, it doesn't sound like something I can trust. It's one thing to say we're having some disagreements about cultural issues, but this cultural war killed a lot of people. And it has not gotten any better, really, since then. So yeah, what do we do about this? If science is no longer seen as a source of information that you can trust for yourself, what do we do, and where else does that spill out? I wrote a book about this, which I'm not trying to do an advertisement right now, but called The Road to Wisdom, which really tries to get even beyond what's happened to science. What's happened to truth? Is there such a thing as objective truth? Yes, there is. And there are real facts that are established, and they don't care how you feel. And it is not okay to say, well, that might be true for you, but it's not true for me because you don't like that fact. But a lot of our society seems to have fallen into that trap. And science has gotten caught in that. I'm a person of faith. What was almost even harder to see was that the group most likely to have distrust of science and to decide not to trust the vaccine were white evangelical Christians. I'm a white evangelical Christian. And as much as many of us tried to explain why, if you're praying for relief from this terrible pandemic, maybe this is God's answer, that didn't play in a lot of people's views. So what do we do about this terrible loss of trust? And it's been so inflamed by the polarization of our society about absolutely everything. And politics has found its way into medical research and medical facts in a way where politics does not belong. And yet many people have found themselves so attached to their particular political tribe that everything else gets filtered through that. And that's not a good place to be when you're trying to figure out what's true about something about science and medicine. I think we can get past this, but we're deep into it right now. I think it's going to be up to all of us. I don't think our politicians are going to solve this. They're more polarized than the country is. 2/3 of us, I think, are in this group that More in Common calls the exhausted middle. And we're kind of checked out because it's just like too unpleasant, all the animosity, all the yelling at each other, all the lack of civility. But maybe we could be part of the solution to that. And I think that means each of us deciding to take on some responsibility to do a couple of things. One is to really reach out again to people who don't agree with you instead of hiding from that. I'm part of Braver Angels, which tries to do this in a very systematic way. And it's been a really good learning experience, bringing together people who are diametrically opposed to their views on something like gun control or immigration or abortion. And you have to listen to each other. I mean, really listen. Listen so well, you can say back to that person, here's what I heard you say. And they have to say to you, that's what I said. How often do we do that? Most of the time, if it's somebody you don't agree with, you're already planning your response. Kind of tuning out whatever it is they're saying, oh, these people don't know what we're talking about. I'm going to set them straight. And that never, ever works. They just get more dug in. So we've got to learn to be better at this kind of communication experience, listening to each other. And I found through Braver Angels, some of the time, after I heard that other perspective, I realized, probably my own view needs to be tempered a little bit here. I'm in my own bubble. I need to get outside of it and see why reasonable people, good, honorable people, are coming up with a very different view about something, even public health issues. We need to do more of that. And the other thing that we've got to do is a better job of figuring out how to apply appropriately our decisions about what information to trust. I think trust comes down to 3 things, but there's an extra one that gets in the way. I think you should trust a source that has integrity, that has competence, they know what they're talking about, and that has humility. They're also willing to say there are limits on what I know. I don't know about everything. But there's another 4th area that now seems to have taken the dominant role, which is that source part of my tribe? Are they aligned with me? That's dangerous. If you're talking about a fact, the tribe shouldn't matter, but it does now. Which means we can be seduced into believing things that really aren't true because it happens to be kind of the group that we're used to hearing from, and it kind of fits with our previous biases, so we just take it on board. And we can reject things that are actually really important for us to know because they came from those other people, and we don't trust them. We've got to get past that. So the book basically suggests that kind of thing. It even has a pledge in it about what each of us might be willing to do if we want to be part of the solution. Stop being part of the exhausted middle, require some energy here, and see if bit by bit we can bring our society back to the kind of place where I think we all want it to be, and it's something you can do as opposed to just feeling hopeless.
I get to make an editorial comment. So one is, I am not positive, but pretty sure that the mRNA vaccination gave us a technology that we're going to be able to use for all these gene correction things we've been talking about. We just didn't think, how do you come up with something that will be applied, scalable to apply to thousands of people Well, that's it. We've shown that it can be done, right? So that's really huge. Another one is one of my other mentors, a woman named Pam Davis. So Pam is the person who hired me. And she said once upon a time to me, she said, yeah, when I went through medical school, they told me that half of what we're going to teach you is wrong. The problem is we don't know which half it is, right? And that's science, right? We're just doing the best we can to figure things out. So, and third is I know all of you can't be at the event tomorrow night, but if you are, that book, Road to Wisdom, is something we're going to auction off tomorrow. And so Francis is not promoting himself, but we're going to have the book, and you will get the opportunity to have it signed by him if you so desire to bid on such a thing. So I get to make those plugs, you don't. So all right, anyway, so that's my my take on things. So any other questions that, yeah, Jeff?
My sense is that the biggest hat you wear is your PhD hat. But as a physician, I'm curious, when you graduated medical school, what you did your residency in, and did you ever practice clinical medicine?
Jeff's an emergency room doctor, so he's...
All right. So I got my PhD first. I thought I wanted to be a chemist. So I majored in chemistry in University of Virginia undergraduate. I ignored life science because it seemed way too messy. It is kind of, by the way, but it's also really interesting. And I went off and got a PhD in quantum mechanics at Yale. But along the way, began to realize I'd missed out on some really exciting things that were happening in life science. I just ignored all of the issues about DNA and RNA and the fact that this is a digital science after all. And I had this crisis about what am I going to do? And I decided I better make a dramatic change and applied to medical school, even though I had not had much interest in that idea until then. I went to the University of North Carolina. I loved it there. It was a great place to immerse yourself in all the aspects of medicine. I fell in love with internal medicine, so I stayed at North Carolina Memorial Hospital, did a residency in internal medicine. I was chief resident in that same hospital in medicine, and then went off and did further training in genetics, but a third of that was clinical, and actually, believe it or not, it was in pediatrics because that's where most of genetics services were in the 1980s. This was kind of scary for an internist to walk into the newborn ICU and be expected to do a physical diagnosis on a subject that was about that big. But I learned a lot in that space. And then, yeah, I ended up at the University of Michigan where I met this guy. And he will tell you that when I was on the attending month, I would disappear doing my thing and ran a clinic half day a week seeing patients with genetic disorders. When I went to NIH in 1993 to run the Genome Project, I had to pretty much give up my clinical activities, although I still occasionally see patients in the NIH Clinical Center who have rare diseases, especially individuals with progeria. And I miss that. I loved the opportunity to work with patients. You will agree with me, I think, that it's a very different kind of experience than being in the lab. Being in the lab, you can go for months without really feeling like something important happened because it's always, I'm on the way to discovering something that matters, and most of my experiments failed anyway. But I don't think I ever remember having an interaction with a patient where I thought that was a waste of time. It's just different. You get a different kind of sense of why you're there.
All right, another editorial comment. So one of my memories of the days back in Michigan was, here's a guy who had to also teach the medical students as part of his job. And he knows full well there's nothing that is more difficult for me than public speaking. So
you're doing okay tonight.
Well, I've had a couple cocktails too. But
and those beta blockers are good too, right?
Yeah, I won't go into that history, but he's he writes prescriptions as well. but watching him, the love for teaching and getting information across, he would walk into his office, and this is back in the day you didn't have PowerPoint, you had 35 millimeter slides that were laying out, so on and so forth. He'd sit down there 5 minutes before the lecture, and he'd pull out these slides and lay them out and so on, and give a talk that looked like it had been practiced for weeks. And it was the most coherent, clear thing and stuff. So anyway, it's part of his magic, his gift. And I was going to attribute that to the thespian background of your mom and dad. Maybe that's not right, but anyway, that was my take on it. So
It did not hurt because, again, my dad being the drama professor, my mother being the playwright, I was shoved on the stage by age 5 and loved it. And it did give you a chance in a small outdoor theater where you could see the audience to know whether you were connecting or not. And when you're talking to medical students who all have their newspapers in front of their face, you know you're not connecting. So you have to pay attention to that. Revise your plan a little bit.
all right, anybody else? Yeah, so Claire? And then John, you're next.
So I know everybody here, well, you're a scientist, you guys are scientists, and this is mostly about science, but I know that patient-centered research is becoming more and more popular, and I'm wondering if you have thoughts about kind of like patient advocacy. You mentioned earlier about, you know, the NIH funding and how like we need to be advocates for that. But I'm wondering if you have thoughts on like patient advocacy and like what are the most meaningful things patients can do to further science as lay people, not scientists.
Oh, I love the question. Everything I think I've worked on as far as a medical research issue has been so benefited by having patient perspectives. And it does help shape the direction that you take. This progeria project I mentioned earlier, on our little team of 4 people, one of them was a 28-year-old with progeria, the oldest, a young man with a molecular biology degree, Sammy Basso, who I'm sorry to say we lost a few months ago. It made such a difference in the way in which every Monday afternoon, as we tried to wrestle with what's the next step, Sammy would anchor us. We need that. Right now, as I think American biomedical research is really in a crisis, I don't think most of the American public knows that. I don't think my complaining about that is going to make much difference to somebody in Nebraska. But I think a patient story of somebody who's in the middle of a medical crisis and is counting on research to be their hope for the future, those stories matter. My passion right now, the thing I'm spending the majority of my time on, is to try to see if there's a way to create a story bank of those kinds of narratives. Authentic, from the heart, straight to the camera, patients who are willing to talk about their experiences and why medical research has really mattered, so that people will begin to see those and say to themselves, wow, that could have been me, or it could have been my kid. Maybe people with cystic fibrosis who can talk about how they are now looking forward to a future they didn't know they were going to have. Or somebody with cancer who had stage 4 disease and figured, well, there's no stage 5, I guess I'm on the way out, and now are cured. And we're seeing that more and more. People with Alzheimer's early symptoms who are counting on that next clinical trial to slow down the process, and those trials are being slowed down right now. Just talking authentically, openly, not politically, about why this all matters and why, of the many things that we do as a culture, this is a really important one that shouldn't be in a circumstance of being so severely cut back right now, which it is. So yeah, there's never been a more important moment than this moment for patient advocates to step forward. I spent a lot of my time talking to those folks. I think they're pretty fired up. We need a way to make that happen and get spread around, which these days is going to be by social media. And I'm learning a lot more about that than I knew before. And if you know anybody who's got a check for $5 million, I know how to spend it.
I don't know that, but I volunteer to be part of the patient stories thing. I don't have a genetic disease, but
Okay, I'm going to follow up. You're going on to my spreadsheet.
that's it. I was working for half at that moment.
So on the trust issue, I don't know how we scale this, but I think being able to be in the same room the way we are now with experts and lay people and have the kind of dialogue we're having now is really a way to begin rebuilding that trust. That's my editorial comment. My question to both of you is, what impact do you think advanced computing technologies like artificial intelligence, like quantum computing might have on drug discovery, drug design, or other areas that you can talk about?
So
Go
I
ahead,
think
Mitch.
there's a lot of different answers to this, but one of them is it allows us to develop hypotheses a lot faster than ever before. Right? I still think there's no substitute for empirical observation. You can do all the AI you want to, but you still have to find out whether or not it's correct. And so that helps us more quickly come up. So I can't scour the literature and hit everything that I need to read, but the computers can do that, and they can try to synthesize that information in a way that I never could. So I think that's useful. But keeping in mind, it's never doing anything by testing. It's only taking information that it's gotten. So I think that's the biggest use in my mind. So
You still got to have experimental capabilities, and that still means doing experiments at the bench. But it is an incredibly powerful tool. I think my lab works on diabetes. I should say my former lab, because I left NIH involuntarily last May, but I still can say I'm connected to the researchers that are working hard there. Diabetes is an incredibly complicated story with hundreds of different genes that can play a small role in contributing to risk. And how does that all work together? It's impossible to sort all that out without some computational help, and AI is really beginning to make an impact there, and that's good. So my lab, which used to be in the past mostly people at the bench, 2/3 of them are now AI experts working on the computer, and that's good. But we shouldn't get too carried away about it. It's not going to substitute for actual experimental data, and it's not going to substitute for human intuition and ability to think beyond what's already known into something that isn't known yet. I'm a little concerned. We're in this hype zone for AI right now that some people might think, okay, we don't need anything else because we've got this. That will not work in medicine. I will say for drug development, you asked about that, the people I talk to who do this in industry, they really are finding it really valuable. Once you know, here's your target. It's a protein of some sort, and you want the drug to stick to it. In the past, you might have to try 400,000 drugs to see which one had the best way to stick. Now you model the whole thing because you can figure out the shapes using the remarkable ability of AI to begin to do that. And so instead of trying 400,000 compounds, you already know somewhere in this group of 100 is going to be your winner, and you do things so much faster.
And John, just for context, so when I started working with Francis, everybody was doing one gene at a time or one protein at a time. That's just the way it was. We did one thing. Now we look at all 20,000 genes at once and a lot of other things at the same time. So now we're relying on essentially what Amazon does to find out what you're going to buy next, what Spotify says you're going to listen to next, all those pattern recognition kinds of things. Those we apply now in biology as well and try to figure out if we can understand what are the patterns that happen with those 20,000 genes. So that's another area. So,
Wearing these people out?
I think so. Yeah, no, this has been very effective.
Diane has a question. Like, when is this going to be over?
I'm going to let you collect your instrument over there. I just want to tell everyone that Francis and I have many opportunities to speak with audiences Tonight is the first time we have met with citizen science fans. You are a different audience to us. And it is incredibly gratifying to be in your presence. This is what our country needs. Thank you very much.
I can't tell you what it means to me for you to meet my friends. So we're going to wrap up with something that I think will be a different experience for everybody, right? So I don't know what you envision a bunch of science geeks doing and so on and so forth, but I want you to leave thinking that, hey, they're real regular people just like us. So at the Cystic Fibrosis Conference, so here's a scientific medical meeting with about 5,000 people. Usually, you're getting these highly technical presentations, and then this guy gets up with his guitar and starts singing. You don't see that at any other venue. So anyway, Francis, I don't know what other introduction I can give you.
Yeah. So it's a blues song. It's got a little 12-bar blues things going on here. And we'll see how this works. Let me be sure I'm in tune. That's not too bad. Disease don't care if you're left or right. Disease don't care if you're black, brown, or white. Disease don't care if you're rich or poor. Disease will find a way to come a knocking at your door. So come on, people, won't you join me, please? Let's get it all together now and knock out disease. You know what you're trying to do, man? Disease don't care if you're old or young. Disease don't care about your mother tongue. Disease don't care about your life's pursuit. You can wear a hoodie or a three-piece suit. So come on, people, won't you join me, please? Let's get it all together now and knock out disease. Let's knock it out. Now here comes the bridge that I just wrote. So some kids and parents carry a real heavy load of the slip in the letters of the DNA code. But if you don't want them to be left in the lurch, their very best hope is health research. We're making great strides in curing disease, but cuts to NIH have research on its knees. If the fight for our kids is gonna succeed, you are the people whose help we're gonna need. So come on, people, won't you join me, please? Let's get it all together now and knock out disease. So come on, people, won't you join me, please? Let's get it all together now and knock out disease. Oh, yeah. Thank you all. What a lovely, wonderful evening.